Gemstone Senior Thesis Conference. My name is Kristen Scandal. I'm the Associate Director of the Gemstone Honors Program, and I have the privilege of working with this wonderful group of students in this Team Brain Blast. I'd like to thank their librarian, who I think is unable to be here today, Annalena Jangalova, and the team's mentor, Dr. Kara Duffy, for working with them since the beginning. This is the first group of teams that I formed in my role as Associate Director, and it's really rewarding and exciting to see where they've come. The team will present for about 25 minutes, and we will ask our panel of experts to offer the first questions, and then we will have some time for questions from the general audience. I do ask that if you have a cellular device to please silence it for the duration of the presentation. And with that, I will turn it over to the Team Brain Blast. Thank you.
Good evening, everyone. My name is Vivinti, and presenting with me today I have Shannon, Caroline, Callis, and Alana. And on behalf of Team Brain Blast, we'd like to welcome you to our thesis presentation and defense. Thank you so much for coming to learn about the project that we've been working on for the past 3 years. We've been investigating the diabetic brain and studying the effects of pioglitazone and insulin on the cellular processes and pathology of Alzheimer's disease. And of course, this wouldn't have been possible without the guidance of our mentor, Dr. Kara Duffy. Alzheimer's disease is the most common form of dementia and is characterized by the loss of memory and cognitive function. Currently, it impacts 5 million Americans and has led to 500,000 deaths per year. And this has made it the 6th leading cause of death in the United States. It can also have a huge mental and emotional impact on not only the patients, but the family and friends of the people who are inflicted with Alzheimer's. And it can also prove to be a huge financial burden. In 2015, $226 billion was spent on Alzheimer's. So clearly these statistics warrant further research into the disease itself. So Alzheimer's is a neurodegenerative disease, which means that it causes neurons to die. So in this image, you can see on the left a healthy brain, and on the right, a brain inflicted with Alzheimer's. And you can see that the Alzheimer's brain is significantly smaller, especially areas like the cerebral cortex and the hippocampus, which are 2 areas of the brain that control for memory and cognitive function. So there are 4 discernible manifestations of Alzheimer's, known as the 4 A's. Amnesia is the loss of short-term and then long-term memory. So for example, someone could forget where they put their glasses, and then in the long term, forget the names and faces of relatives. Aphasia is the loss of the ability to communicate effectively. So someone may not be able to interpret properly what you are saying to them, and then they may not be able to formulate a response. Apraxia is the loss of the ability to perform pre-programmed motor tasks. So someone may not be able to draw, or they may not even be able to walk. And then agnosia is the loss of the ability to interpret sensory signals. So for example, someone may not be able to perceive or process pain, which can lead to unintentional self-injury. So these 4 A's, again, it's important to remember, all center around memory loss and loss of cognitive function. And it's those parts of the brain that have decreased significantly in size in this image. Alzheimer's can't be diagnosed officially until after death, but there are tests that can be performed to ensure probable diagnosis during the lifespan. So there are several risk factors for Alzheimer's, some that can be controlled and some that cannot be. Genetics determines the 2 types. So there is familial Alzheimer's, which is hereditary, and sporadic, which can be based on the presence of certain genes as well as external risk factors, such as lifestyle. But 99.5% of Alzheimer's cases are sporadic. Age can also play a role in the manifestation of Alzheimer's, and 96% of patients are currently over the age of 65, and that risk doubles every 5 years after the age of 65. Lifestyle factors such as smoking, obesity, and lack of mental activity can also play a role in the manifestation of Alzheimer's. But the risk factor that we are looking at with our project is the comorbidity between Alzheimer's and type 2 diabetes. Alzheimer's and type 2 diabetes are both involved with deficiencies in glucose uptake. And in this image, you can see on the left a healthy brain, and on the right, a brain with Alzheimer's disease. The red represents glucose metabolism, and you can see that on the left in the healthy brain, there's a lot more glucose metabolism happening than in the Alzheimer's brain. So between Alzheimer's and type 2 diabetes, the location of glucose uptake deficiency does differ, but researchers have found such a strong link between the 2 diseases that Alzheimer's has even earned itself the name of type 3 diabetes. And this is the lens through which we are viewing Alzheimer's disease. I'd now like to pass it on to Shannon to talk to us a little bit about the biology of Alzheimer's.
So there are main 4 pathologies of Alzheimer's that we were studying in our project. The first being amyloid plaques, then tau tangles, chronic inflammation, and diminished cerebral glucose metabolism. All of these pathologies contribute towards Alzheimer's being a degenerative disease, and that each of these pathologies contributes towards widespread cell death in the brain. The first pathology is tau tangles. Tau tangles disrupt the intracellular mechanisms, or inside the cell mechanisms, and they disintegrate the microtubules, which are responsible for structure and transport throughout the neuron. You can see in this image on the right how tau tangles contribute towards this. The next pathology is amyloid plaques. These plaques are made up of a toxic form of an otherwise normal protein called amyloid. This toxic form can be thought of as sticky and causes other sticky amyloid proteins to aggregate into these large extracellular or outside the cell plaques. These plaques get in the way between neurons and disrupt neuron-to-neuron communication and cause a trigger for programmed cell death. And then we have inflammation. Microglia are some of the immune cells in the brain. They are normally able to digest small amounts of plaques. However, in Alzheimer's, when there are a very large number of plaques, the microglia are overwhelmed and not able to digest all of them, and they call for more microglia, which yields to a prolonged immune response. Normally, a little bit of inflammation is very normal and healthy for the brain, but this chronic inflammation actually leads to even more brain damage. The last pathology of Alzheimer's that we were studying was diminished glucose metabolism. When you eat, to get energy from the food that you eat, you need to get glucose from your bloodstream into the cells where it can be metabolized for energy. This process is regulated in the body by the hormone insulin and moved into the cells through glucose transporters known as GLUTs. Both type 2 diabetes and Alzheimer's are associated with insulin resistance, which means that the insulin, as shown on the left, cannot easily bind to its insulin receptor. This insulin resistance in Alzheimer's has been studied extensively, especially in a very interesting new study called the SNF study, which stands for Study of Nasal Insulin to Fight Forgetfulness. This used a nasal insulin spray in humans with Alzheimer's disease, and it was actually found that this insulin improved the memory of the patients. So to look at how Alzheimer's has diminished glucose metabolism, we can more in depth explain how glucose uptake normally happens in the brain. So the brain has very high demand for glucose, and this is because your brain is constantly working, whether it's through regulating your breathing or your pulse. So having insulin limit the amount of glucose that could get into your brain would not make a lot of sense. So the brain actually relies heavily on insulin-independent glucose transporters, GLUT1 and GLUT3, for example. On the right here, we have a very simplified model of a neuron that has the glucose transporters GLUT1 and GLUT3, but no insulin or insulin receptor. To get glucose from outside of the cell to inside of the cell, where it can be metabolized for energy, the glucose can be taken in through glucose uptake, which here is very simple, just goes through the transporters. The comorbidity of Alzheimer's disease and type 2 diabetes, however, suggests that there is a similarity in the way that the two diseases intake glucose. So now we can look at how insulin plays a role in glucose uptake. This is actually interesting because insulin, even though the brain is mostly insulin independent, it does have some GLUT4 in the brain, especially in the hippocampus, which is an area of the brain that focuses heavily on memory. And this is actually the first place that you can see amyloid plaques begin in an Alzheimer's brain. And it is also where we focused our study. So again, we have a very simplified model of a neuron, but in this case it has GLUT4, which is insulin dependent, and insulin and its receptor. When insulin binds to the insulin receptor, it signals something called the insulin signaling pathway, which is all of the steps between insulin binding to its receptor and moving GLUT4 to the plasma membrane so that glucose can get into the cell through glucose uptake. To further study the insulin signaling pathway, especially in Alzheimer's disease, we used a type 2 diabetes drug called pioglitazone, which we'll now refer to as PGZ. And this drug enhances glucose uptake. As you can see here in this diagram, PGZ actually stimulates part of the insulin signaling pathway. And this stimulation results both in more GLUT4 and more GLUT4 moving to the plasma membrane, so that even more glucose can enter the cell, and this causes increased glucose uptake. So to take a closer look at PGZ, we can see here on the left that PGZ is a very common type 2 diabetes drug prescribed under the name Actos. It contributes to reduction in Alzheimer's pathology in a few ways. It can help clear amyloid plaques, prevent tangle formation, reduce inflammation, and as we saw, increase glucose uptake. PGZ, therefore, should supplement the effects of insulin on Alzheimer's disease, which we've already looked at. However, previous studies using PGZ in Alzheimer's disease have only ever administered PGZ orally, and this is important for a reason that we will now look at. The blood-brain barrier surrounds the central nervous system and the brain and filters unwanted or harmful molecules from entering the brain. When administered orally, only 18% of PGZ is actually able to pass through the BBB and get to where the Alzheimer's pathology is, the brain. So intranasal drug administration is a technique that allows molecules entered through the nose to actually bypass the blood-brain barrier and go through the nasal membrane, as we can see here on the right. Nasal spray enters the nostril and then moves to the nasal cavity, where it then is able to cross through both the olfactory nerves and the olfactory bulb and move directly to the brain. This would be an extremely useful application of PGZ, but it has actually never been done before, which brings us to our research of using intranasal PGZ, which Caroline will now explain.
We sought to address 3 research questions. Does intranasal administration of PGZ and insulin sensitize insulin receptor signaling to increase glucose metabolism? Will long-term administration of PGZ and insulin treatment reduce Alzheimer's pathology? And do reductions in pathology persist after completing the drug administration? We hypothesized that PGZ would cause reduced Alzheimer's pathology, such as plaques and tangles, long-lasting effects on pathology, and this would be related to an increased glucose uptake in the brain. In order to address our research questions, we begin with a pilot study in order to determine the optimal PGZ dose. We followed this with our main study, which had 3 phases, each addressing one of our research questions. To conduct our research, we used a mouse model, the triple transgenic mouse, which is genetically modified to have hereditary Alzheimer's. There are 3 genes changed in this mouse to ensure that they will develop Alzheimer's pathology, and they develop this pathology around 1 year of age. The post-mortem characteristics seen in these mice are similar to humans, so we can use research on these mice to help understand Alzheimer's disease in humans. So we used a novel method to apply our intranasal PGZ, which is more direct. We dissolved PGZ and insulin into saline solution, which we administered to the mice. We used an intranasal grip to restrain our mice during dosing, rather than sedating them, which reduced stress for the mice during treatment. As you can see in this video, we are administering the dose of PGZ to our mice. After restraining the mice, we use a syringe to put droplets of solution onto their noses, which go directly into the olfactory up to the brain. We alternate between nostrils as we dose them. This method is similar to a nasal spray that humans would use. We began with our pilot study in order to validate the intranasal administration technique, as well as determine our PGZ dose. In order to come up with a reasonable estimate for the dose, we took into account the standard oral PGZ dose, the brain metabolic rate, and the body weight of our mice. This led us to a number of 30 micrograms, which we halved and doubled in order to test in our pilot study. The pilot study used 8 non-transgenic mice split into groups of 2, which received 0, 15, 30, or 60 micrograms of PGZ. After 24 hours, we collected samples and sacrificed the mice. In order to understand the relationship between PGZ and glucose metabolism in the brain, we injected the mice with a carbon-13 labeled glucose tracer before sacrificing them. This is a special type of glucose which allows us to measure levels of metabolites in the brain. We use a process called fluxomics to analyze the levels of these metabolites in our samples. In the graph, you can see the ratio of metabolites, serine, aspartate, alanine, and glutamate for each dose of PGZ. As you can see, the rate of glucose metabolism, as indicated by the levels of carbon 13 seen in the metabolites, increases with the dose. Thus, we selected 60 micrograms as our optimal dose. There are two possible mechanisms for why increased glucose metabolism aids Alzheimer's disease: the direct and indirect methods, which Kellis will now explain.
As Caroline just said, there are two reasons that we are seeing diminished glucose uptake with Alzheimer's disease. First, we have the direct mechanism. Here, we are proposing that the problem originates in the insulin signaling pathway. Now, there's an upstream mechanism in the insulin signaling pathway that isn't functioning properly. And the result of this is that the glucose uptake in GLUT4 is compromised. We propose that the increased glucose uptake we saw as a result of our PGZ treatment indicates that PGZ is restoring function to the insulin signaling pathway. Next, we have our indirect mechanism. Here, we believe that the insulin-independent glucose uptake mechanisms are damaged. And we also are proposing that our PGZ treatment is then compensating for this by increasing the glucose uptake in the insulin-dependent glucose uptake mechanisms. So these dependent mechanisms are then compensating for the independent mechanisms. So we learned a lot from our pilot study, and now I'm going to talk to you about how we set up our main study. Our main study was broken up into 3 phases, with each phase trying to answer one of our 3 research questions. Also, each phase used 15 mice, with 5 mice in each of our 3 research groups: saline, insulin, and PGZ insulin. While our pilot study used normal, non-transgenic mice, our main study used triple transgenic Alzheimer's mice. First, we have our short-term study, which is called single dose. With this study, we dosed all of our treatment groups with 1 dosage level, the level that we determined from our pilot study, and then samples were collected the next day. This study helped us verify that our Alzheimer's mice have the correct Alzheimer's pathology. Next, we have our long-term immediate phase. This phase is called long-term because we dosed the mice every day for 3 weeks. And it's called immediate because samples were collected 24 hours after our final dose. This test gave us our first look at how our treatments impact pathology in the long term. Last, we have long-term delay. Similar to the last study, we dosed the mice daily for 3 weeks. But it's called delayed because we waited 2 weeks after the final dose to collect samples. This test is important because it helped us see if the effects of our treatment are persistent over time. Oh, wait. So, once we collected our samples, we used immunohistochemistry tests. Now, immunohistochemistry is a way to stain specific proteins. And I'm about to show you some of our actual samples and that were stained with these antibodies. First, we have amyloid. Next, we have our activated microglia, which is an indicator of inflammation. And last, we have tau tangles. Now, with all of these images, the upper image represents lower staining. Now, you're going to see a lot of graphs in these next couple minutes, and it's important to remember that the y-axis scales, this is the counting that we're looking for, because the upper 3 graphs indicate have almost no staining, and the lower graphs, indicated by these brown widespread splotches, have high staining. So we use these immunohistochemistry tests on our single dose test, which, as a reminder, was how we first verified that our Alzheimer's mice have Alzheimer's pathology. We found that across the 3 tests, the results were consistent and the mice did have the pathology that we were looking for. So this was our short-term test, and now Alana's going to talk to you about our long-term tests.
So all the results from our long-term media and delay tests are a reflection of the immunohistochemistry that Kelly just explained. Now I'm going to orient you to this amyloid plaque graph because the following graphs with the tau tangles and inflammation have the same layout. On the y-axis, you'll see the quantification of the pathology we're measuring. So in this case, you see the amyloid plaque count. On the x-axis, you see our treatment groups, saline, insulin, and PGZ plus insulin. You'll also see that we'll have two graphs juxtaposed together. The left side will always be our phase of long-term immediate, and the right side is our phase of long-term delay. Now before showing you our results, we want to remind you of what our hypothesis would look like on the graph. So this white line represents a trend of what we would expect. We would like to see that the PGZ plus insulin group shows the least amount of pathology. The insulin group has a middle amount of pathology, and the saline group should have the most pathology. Now here are our results, and also some results of our statistical testing. So the letters represent the groups that are statistically the same or different. Let's look at the left graph that's red. If you look at the saline group, it has an A, and if you look at the PGZ plus insulin group, it has a B. If the letters are different, that means the groups are statistically different. So we can see that the saline and PGZ groups are the only ones that are statistically different for the long-term immediate. Looking to the right at the long-term delayed, there's actually the same pattern of significance where the only statistically significant different groups are the saline and the PGZ plus insulin. Now statistics are important, but it's also important to look at the greater data trends that we find. So we determined that these were the trends that were applicable for this staining. The long-term immediate was what we hypothesized, but the long-term delay was not. Now to keep track of what we've been getting from these graphs, we have this image that we're going to fill in. So we can say that the immediate phase did reduce amyloid plaques, but we did not see this in the delayed. Moving on to our tau tangle pathology, the only difference with this graph is that the y-axis is tau tangles. We hypothesized the same trend for our treatment groups. Our results, you can see, don't have any letters on them because we didn't see any statistical difference between any group. As you can see with our trend arrow, we also don't see any trends. To take this tally for our helpful diagram, we can say that we did not see a reduction in tau tangle formation as a result of PGZ, and we did not see a delayed reduction. And now for our final graph, inflammation, where we have microglia ranking on the y-axis. Again, we have the same hypothesis. Here are our results. You can see that for the long-term immediate on your left, that the saline group was statistically significantly different than insulin and the PGZ plus insulin. And in the long-term delayed on the right, you can see that the PGZ plus insulin group was the same as the saline and the insulin groups, but the saline and insulin groups were different. From this, we got that our trend for the long-term immediate matched our hypothesis, but the trend for the long-term delayed did not. Keeping up with our tally, we'll say that with the reduced inflammation, it worked in the immediate, but in the delayed, it did not. So now we're going to discuss why we think we saw the results that we did. So we expected an immediate reduction in amyloid plaques due to the PGZ, because we know that PGZ actually helps degrade the plaques, and it also helps keep them from being formed. For the reduction of inflammation, we know that PGZ prevents activation of microglia, and microglia are the marker of inflammation. In addition, inflammation responds to the plaques, and by having fewer plaques be created and more being broken down, we would see less inflammation. We expected to see a reduction in tau tangle formation because we know that PGZ should inhibit the formation of these tau tangles, but it didn't happen. We think that the nature of our mouse model could have had slightly different tau pathology than what has been done in other literature. We also think that a higher dose of PGZ might have been needed for this specific pathology. Now to address all of our delayed results not being what we expected, it could be that PGZ just leaves the body too soon to see long-term effects, and it could be that a higher dose than 60 micrograms would be needed to see any effect at all in the long term. So pulling it all together, it's great that we saw any statistical significance since we only had a number of 5 for each group. And we think that with a larger sample size, we would have more statistical power and we could see more statistically significant results by doing the same things that we did. We would also like to further investigate fluxomics results for our second and third parts of the study so we can look at what's actually happening with glucose metabolism with long-term treatment. In addition, we think we could further optimize PGZ dosing. We did this with a pilot study where we found out that 60 micrograms is ideal, but it seems like an even higher dose would be appropriate. And also we used a saline solution to deliver it, but because that's what has been used with insulin, but maybe a different vehicle would be even better for delivery. So we would like to thank all of you for coming and supporting us today. We have some special acknowledgments to make, including we'd like to thank Dr. Brian Duquette and Dr. Kara Duffy as our mentors. We'd like to thank the Gemstone staff, Dr. Kostonwick helping us out, our librarian Madalina Chivalova, the Animal Sciences Department, Dr. Lee, Dr. David Zimmer, the longevity donors, which a lot of you are sitting in this room. We give a special thanks to Chris Buck and Robin and John McHugh for their donations. Our discussants, the Donald and Catherine Weston Award for Research Excellence presented by the University of Maryland Libraries, and our family and friends, which most of you are. So before we move on to questions, we would like to say a few words about our late mentor, Dr. B.
As some of you may know, our mentor, Dr. Duquette, sadly and suddenly passed away early of our junior year. I'm now going to share with you some words that we as a team collectively put together in memory of Dr. We were the last Gemstone team of our cohort to receive a mentor because it was difficult to find someone who specialized in our research area of interest. Although he did not have much background in our research area, Dr. B stepped up and offered to be our mentor because he had such a great time mentoring a previous Gemstone team and was excited to learn along with us. Dr. B was integral in the development of our project. He was a brilliant scientist, excited to learn about the new area. And also, he was an incredible mentor figure as well as friend to each and every one of us. He showed genuine care and interest in all of our lives and also our academic endeavors as well as extracurricular interests. He also liked to share gossip with us, and we would often have to focus him at our meetings to talk about science. It is clear that Dr. B made friends everywhere he went because many of his colleagues were happy to help us as a team. It was because of Dr. B that we were able to meet our current mentor, Dr. Kara Duffy, who had agreed to be our team expert in the beginning stages of our project. In dealing with Dr. B's passing, our team again found ourselves lucky enough to have someone step up and be our mentor, even when the circumstances were not easy. And for that, we are incredibly grateful to Kara. We could not have achieved finishing our research without her or anyone else who has helped us along the way. And the fact that so many of Dr. B's colleagues had gone out of their way to help us after his passing is a true testament to his character and the genuine relationships he had with them. In team meetings, we often reminisce about our memories with Dr. B, or ask, what would Dr. B think about this? It is clear that his presence still remains all throughout our project, and for that, we dedicate our research to Dr. Brian Duquette. We hope we make him proud. Thank you.
Thank you, well done. Now I have questions from our panel of discussants, and then we will have time for questions from the audience. And as each discussant, you just introduce yourself before you ask your question, and we'll go down the road.
Hi, my name is Bill Reginald, and I'm honored to be here. It's a very nice presentation, thank you. And I was really impressed with how much you took on. The experiments you did really are beyond what you usually see written up in any given page. And, you know, as a result, I think not everything went perfectly, which I think is to be expected. So one question I had is, what, and I'm not sure who to address this to, but you can decide, what do you think happened with the fluxomics experiments? I understand that somehow you didn't get any data from the sample. I just wondered, in particular, and this is kind of a technical issue, but were there no peaks coming off the column, or does anyone want to elaborate on that? Any ideas what went wrong? Just because I think it's important to figure out if you were to do it again, what you might do differently.
So we actually ran some fluxomics results from some of our previous, on some of our long-term studies. However, due to logistical issues, such as a computer dying, we couldn't actually physically get the results. So there were some issues with that. We did run the fluxomics data with our short single dose test. However, we didn't perform the proper standard for the test that we were running, and so we were actually unable to locate any significant results looking through the entire fluxomics run that we did. We were unable to determine where in that data we would actually be able to find anything related to our glucose testing.
And did you have help with the analysis, or?
Yes, we did have help from Dr. Yuan Li with that. It's something we actually are looking to continue pursuing. We're actually trying to fix it after this presentation.
Thanks.
My name is Nam Wang. I'm from Chemical and Biomolecular Engineering here. The delivery was very smooth, I thought, and very professionally done. And I also like the introduction, which helped us understand what the problem is. And okay, let me ask a question here, since I was asked to do that. Actually, I have more than one, of course, but anyway. For example, in the summary you investigate 3 different things, and in your introduction you put a lot of emphasis on glucose uptake, right? And for some reason that wasn't measured or mentioned. Why did you not look at glucose uptake? Because I thought that was one of the main things that
Okay, so the question was about why we didn't have an emphasis on glucose metabolism for our later studies that we were talking about, other than the pilot. So it's actually a similar answer to the first question. So we did have some technical issues with the fluxomics that we were planning to run. So we didn't get the results that we expected because we were planning to look at glucose metabolism through the fluxomics analysis. So that's really the main reason we didn't have that.
Hi, I'm Ricardo Araneda. It was a really great presentation. One question regarding the model. We're using this, already was a 1 year old. You said that you were using your mind because that's when they started seeing the symptoms. Do you think this treatment would have been different if you had it done before? Like before they started showing the symptoms?
So that's a really interesting question. It's one that when we were developing our project, that's a question we really wanted to answer, but we realized we already took on a lot and we couldn't take on all of that as a project too. So in the literature, a lot that we've seen with insulin showed treatment once Alzheimer's had onset. So we wanted to mirror that with PGC, and in addition, I just had the other idea. So that's the main reason. And we wanted to make sure that they would develop Alzheimer's, so with the 1 year thing, we wanted to be certain that we were treating it the same with all the different.
Hi, I'm Matt Resch, faculty in psychology. It's really terrific. So I guess my question kind of gets back to what you just talked about, developed Alzheimer's. So you have all these wonderful pathologies in the brain that tell you that this looks like an Alzheimer's brain, but ultimately it should be based on the behavior of the animal. I was wondering if the knockout showed behavioral deficits, or if you noticed any differences between them, or how this kind of relates back to your introduction where you laid out the 4 A's of Alzheimer's.
So at one point of our study, we were actually planning on conducting a Morris water maze with our mice in order to evaluate their behavior changes. We were not able to do that, so we just looked at their cellular pathologies, but that's definitely something that we considered and would like to explore in future directions.
We do have some time for questions from the audience. I'd like to open up the floor. Your section leader.
So my question is two-part. So when you were determining the oral dosage for the PCG for the rats, did you use a formula, like was it by weight of the rat or anything like that? And then afterwards, if you were to determine a higher dosage, have you thought about what that dosage would look like?
That's a good question. She's my So it was a fun challenge to calculate that dose. So the way we did it is there has been research with oral PGZ on mice for Alzheimer's. And there's been research on insulin orally and insulin intranasally. So kind of compromising, we looked at metabolic rate, we looked at the mass of our mice, and we came up with that calculation of 30 micrograms. And with all those factors, we were fairly confident about it. I think testing a higher dose would be interesting. How high would we go? I think another way this research to go would be some in vitro testing where we maybe find a dose response curve and see where it levels off. We definitely want to not see adverse behavioral effects because it's such a high dose of something, so we were afraid of going too crazy, which is why we doubled it. But I think it's a really interesting area of research that should explore more.
Hi guys, once again, awesome project. I like the progress you guys made throughout all the years. My question is about the intranasal doses that you guys applied. I know that you experimented and looked at the mass of the drug that you used. Did you also look at the concentration and the volume which it was associated with? Did you just look at that?
So the concentration was actually something that we struggled with because some of the PGZ did come out of solution. So an increase in the volume might reduce that concentration, which might allow the PGZ to more effectively enter. But just because of time and essentially there's only so many mice that we had to constrain ourselves with what we were able to test and what we weren't able to test. So we went with 24 microliters as our dose, or as the volume that we administered the doses with, because that was the most common in the insulin research.
Other questions?
I'd like to first of all compliment your team. Well done. You alluded to an alternate way, form of dosing. Can you elaborate on that? The ideas you had, possibly had on that, rather than just through the nasal cavity?
So for the two, there were essentially two types of dosing that can be considered. There's oral dosing and there's intranasal dosing. And actually PGZ, which is the drug in question, had been dosed orally before. But we thought that intranasal dosing might be more appropriate, essentially for two reasons. Because one is that PGZ can have detrimental effects in the body if it's at too high of a concentration. And that two is that only 18% of the PGZ that enters the body is actually able to pass through that blood-brain barrier into the brain. So we thought that the intranasal delivery method was a way to get a higher percentage of the PGZ into the brain as opposed to into the periphery where it could have those adverse effects.
First of all, congratulations. It was a challenging project that you all did well. I had one two-part question, then one comment. One question is about the images you showed in immunohistochemistry tests. So was that, it's like part of the brain that you see through microscope and then take pictures? I just wanted to know how you do that. That was my first question. Second question was, all the y-axis you showed on your three results, so like plaques, for example, you see counts on y-axis. How, so do you actually count how many you see visually, or so how do you do that? And I'll do the comment and then you can answer. Not as recently last week or so, there was a big Alzheimer's disease conference here in DC for policymakers that researchers were coming together and saying that the funding should be doubled for Alzheimer's research. And so if you're really interested in pursuing this, this is a great topic and it's very useful for national health and economic health. So,
So this was about, I'm just going to go back to that slide. So the first, can you repeat the first part of the question?
The first one was the images that you showed. So is it us, the brain that you're looking at?
Yeah, so we took the brains from the mice, and you can freeze them and then slice them into like 24 micron slices, and then those are put onto a slide, and you can run various chemicals over them, and then that is what produces the staining. So the way we quantified the staining was different for the different antibodies. There we go. So for the plaques, we actually did count each plaque, but that was not so feasible if you look at kind of what the microglia and the tangles look like. So what we did is we actually used a Likert scale to rank from low to high on 3 points how severe the pathology is. So the top two pictures are an example of a low ranking, and then the bottom two are high, and we also had a medium. So we had every member of the team rank those two because we know that it's kind of variable, those qualitative analyses, but we had fewer people get trained and specialized to actually count the number of plaques, and that's what gave us our y-axis for that.
Okay, time for one more question.
So I was interested in your long-term immediate and your long-term delayed results. How did you come up with 2 weeks as the delay period? And do you think your results would have been different if the delay period had only been 1 week?
So one of the reasons we selected the delay period of 2 weeks, one was feasibility on our part. We only had so much time in a semester, and we were dosing mice every day, and we only had a little bit of time before finals to collect all of our samples. So that's one reason why, but also with working with Dr. B to design the study, he helped us find some sources in which 2 weeks was one of the tested timelines of data collection afterwards. And since that seemed to be the most feasible for us, that's what we went with. Based on our trends that we did see some immediate effects, I would hypothesize that if we would see better effects at 1 week after rather than 2 weeks, because it doesn't look like these effects are long lasting, but further research would need to be done to determine that. We can only hypothesize based on the trends we see now.
Thank you very much. Please join me again in another round of applause.